Rob Wright is going to start our discussion. And Rob, you're welcome to start your slides. Rob is a science informationist at the Welch Medical Library at Johns Hopkins University. He provides embedded librarian services, including expert searching of literature and bioinformatics databases, citation management, and instruction in workshops and departmental courses. Rob has developed the search strategies for numerous systematic and scoping reviews and assists researchers with mining data from public repositories of biological data. He supports the basic science departments of the Johns Hopkins School of Medicine and the Bloomberg School of Public Health. So Rob will be speaking to us this afternoon about the elements of a systematic process for literature searching, a 3Rs-based example. All right, Rob.
Thank you, Christina. So as Christina just mentioned, I'll be talking about a systematic process for literature searching and use a 3Rs-based example and use it in a commonly used and freely available biomedical database, namely Pubmed. So, sorry, there we go. So why a systematic process for literature searching? For one, it improves search comprehensiveness. And as a consequence, fewer relevant studies are missed. Provides a fuller picture of available methods and findings, for instance, novel 3Rs approaches. There's less of a chance for unnecessary research duplication, furthering the goals of reduction. Provides a measure of confidence in search rigor, and can be used at different points in the research life cycle. So that could include the idea generating stage or the formulation stage of a project, or the funding stage where you need to show evidence of why your project should be funded. And it's also useful as the cornerstone for different types of reviews, ranging from narrative reviews to scoping and systematic reviews. So the sources for this process and the sort of the 3Rs specific guidance that I'll cover today come from searching guidelines from Cochrane and Ural-ECVAM, as well as the projects that Christina mentioned in my introduction about the systematic and scoping review projects that I've worked on. Those have been largely through the Evidence-Based Toxicology Collaboration and the Center for Alternatives to Animal Testing at Johns Hopkins. And in addition to those two sources of background, I've also taught these kinds of skills, which is always useful and revealing about the skills, and you learn a lot. And specifically, I learned a lot teaching in a 3Rs-based course at Johns Hopkins taught by Catherine Herman. So this is the process itself. It's 8 steps. And starts with an initial step of identifying what are the components of your search strategy or your concepts. Follows that with actually generating terms from controlled vocabulary. In the case of PubMed, the mesh database is used to generate mesh terms. And also an important step is finding keywords or natural language terms, also known as text words, to supplement those mesh terms. Applying the appropriate syntax and formatting is another step. Follow that by applying Boolean operators to indicate to PubMed the ways that you want to combine the terms that you're including in your strategy. The sixth step, which is actually assembling and running the search using advanced search features in Pubmed. Step 7 is assessing search quality. And step 8 sort of naturally follows from that assessment because typically in that assessment step, you notice terms that you hadn't included, or you notice maybe terms that are giving off-target results that should be removed. Lots of different things can come out of that assessment, and those things you notice then get built into the next iteration of the search strategy. So I'll be talking briefly about each of these steps in my talk to give you a flavor of how this process can be sort of implemented in the real world using a specific example. And along the way, I'll be highlighting certain aspects of this process relevant to the 3Rs. So step 1 is identifying key concepts. So we'll be talking a bit about how to come up with concepts and how to do so carefully. Documenting your search strategy is an important searching process, much like maintaining a lab notebook for your searching process. And then what does this document look like and how to create one? So this slide is highlighting what I'm calling important terms for searching in a research question. So what's critical to this whole process is to have a clearly articulated research question. And what I have here is the question we'll be working with today. What in vitro studies examine changes in serotonin-mediated neurotransmission in major depressive disorder? So this is our question. It's always nice to have this front of mind to make sure you're staying on track with your search strategy. You're not sort of veering off into territory that's not relevant. And so that's important in and of itself to sort of know what it is you're trying to ask. But in addition to that, you can mine the research question for the major ideas or main themes of your question, and those main ideas become the concepts and the framework around which you'll build your search. So in this case, important terms that will end up being translated into search concepts are in vitro studies, serotonin, and major depressive disorder. And what typically happens in a animal alternative search, at least in my experience, is that there's a study type component of your search strategy. In this case, it would be in vitro studies, sort of stressing the alternatives methodology. And in this case, we have also a biological agent mechanism term or theme or concept in serotonin, and then a disease state with major depressive disorder. What I'll highlight here as non-important terms from your research question include the words changes and neurotransmission. So the research question is important for other reasons. So it is important to say that you're interested in what changes you're observing. It might also be important to stress that you're interested in neurotransmission, but these are not essential elements for the search. So this sort of points up the care needed to come up with search concepts. And a good rule of thumb is to think of what are the essential elements of your research question, the necessary ones that must be in the articles you retrieve. And also, I think it's important to stay away from sort of ill-defined or hard to capture states. So you could have a changes a component to your search, However, it's sort of hard to capture change. Lots of people refer to change in very different ways. There's no sort of standardized way necessarily to talk about change. And even it could be the case that change isn't even mentioned in the title of the abstract. So I'm excluding changes. I'm also excluding neurotransmission as something that I feel like is probably embedded in the serotonin term. So leaving that out as well. But having the research question there, you can always circle back and maybe include a neurotransmission concept if you find that you're being overwhelmed with papers on serotonin major depressive disorder that are in vitro in nature. So what comes out of this process of looking at the research question are three main concepts. Serotonin, or in vitro studies, serotonin, and major depressive disorder. And I number them just as I'll demonstrate later. It's sort of a handy way to just have the concepts, have a shorthand for the concepts, and then indicate how you're going to combine them in your search strategy. So this is the sort of first step, is designating the main ideas from your research question and making them the components of your search. So I like to do this in a Word document. I've also done it a bit in Excel, but I find Word is easier to modify and change. So I sort of tend to use Word to do this. And what's nice about doing it in Word is you're able to record your research question at the top, so it's always there for you to know what you're doing or what you're looking for. You can use it to separate out your search concepts and provide space to build lists of terms underneath those concepts. You can then document different versions of your search. So in that 8th step of the process where you're having different iterations, you can keep track of what changes you've made, how you've combined concepts, how you've removed or added terms. And this is just important because you may end up wanting to circle back to previous versions. It's important, I think, also to have a notes section in your documentation, trying to sort of remember terms and do things on the fly. It's easy to forget what terms you've tried or what terms you've removed. So having a place in your documentation to note those terms, to say term X gives off-target results, consider removing, or term Y was one I found in those search results, test that in the next iteration of the search. Yeah, so my 6th point here is that in a notes section, you can keep track of terms to test in future versions of your search strategy. So this is a sort of mock-up of what the search document could look like. You've got the research question at the top, and then your 3 concepts with space left for populating with terms. So the next 2 steps are actually finding the terms themselves. So what you're going to put in those spaces. We're going to start with mesh terms and define what they are, how to find them using the mesh database, using the Pubmed search builder, which is a sort of built-in way to format mesh terms correctly, and then how to just add these mesh terms to your search when you find them. So what is mesh, or medical subject headings? First of all, I'll say that really they should be called biomedical subject headings because they cover the full breadth of what Pubmed covers, which is biomedicine. So Pubmed, of course, covers disease states and treatments, drugs, that kind of thing, but it also covers genes and proteins and biological processes. So it's really best to think of MESH as this set of terms describing everything that is in Pubmed. Yeah, they're standardized in nature and uniformly applied by Pubmed indexers. So there is a cadre of subject matter experts at the National Library of Medicine who are looking at all the articles that come into Pubmed and using this code book of the medical subject headings to describe the main ideas in the papers. This is also referred to as a controlled vocabulary. You might be familiar with other controlled vocabularies like the APA thesaurus used by PsycInfo or MTree used by Embase. They're all controlled vocabularies, have some similar features, including a hierarchical structure. So this vocabulary has not just a set, it's not just a set of preferred terms, but it's a set of relationships between terms showing how they relate to one another, with more general terms being at the top of these hierarchies or trees and more specific ones below. And it's a system designed to capture a variety of subjects, covering all of biomedicine, no matter how they might be referred to in the literature. So another way to think of MeSH terms is sort of umbrella terms that translate the heterogeneity of how things are described in the literature into a single standardized term. So for instance, the serotonin MeSH term captures references to serotonin, but also 5-hydroxytryptamine, hippofane, enteramine, 5-HT, et cetera. So all the synonyms that folks use to describe serotonin would be captured by the MeSH term. So a little bit about MeSH and the 3Rs. What you see on the right-hand side are some hierarchies or trees from the MeSH database for a variety of relevant sort of animal alternative terms. And as you can see, these are sort of spread out across multiple hierarchies or trees. In vitro techniques lives under the investigative techniques heading. Organoids is under tissues. Animal use alternatives is also under investigative techniques, but sort of separate from in vitro techniques. And then lab on a chip devices is sort of in this microelectronic mechanical device type tree. So you have to, sort of one general rule is you have to do quite a bit of digging to find all relevant MeSH terms for a 3Rs-based search. And then I think it's also important to note that some animal replacement terms that are commonly used or relatively commonly used in the literature don't have any kind of direct MeSH equivalent. So there is no MeSH term, for instance, for microphysiological systems or for neurospheres. They sort of fit into like microphysiological systems, fits under lab on a chip devices, and neurospheres, I think maybe fits on one that's not listed here, which is cellular spheroids. So it can take some time to sort of find or navigate through MeSH to find appropriate terms. But as I'll sort of talk about a bit later, it's really worth it to do so. Animal use alternatives, want to also note as a MeSH term, was only introduced in 2001 and is currently only assigned to about 4,000 articles in Pubmed. So that's also somewhat of a limitation, how articles are actually described as animal use alternative. And then certainly came out in the class that I taught that studies have really oftentimes a mix of different approaches or techniques that they use. So in vitro and in vivo techniques might be used in multiple articles, so that you have to do quite a bit of screening of your results to determine what's going to be relevant to your research question. So why use these MeSH terms? They make your search better because they're the preferred terms that Pubmed uses. So definitely tapping into the ways that Pubmed describes articles is really powerful. They also automatically search for more specific terms in the hierarchy. So based on those hierarchies you saw in the previous slide, everything below what was in bold is going to be found if you use that sort of parent MeSH term. The parent MeSH term will find all the child terms. And then also mesh records include lists of synonyms that can be used as keywords, which we'll talk about in a bit. So the mesh database is the tool for finding mesh terms, and it's relatively easy to find. It's on the Pubmed homepage under Explore. There's a link to it there. You can also just Google the mesh database. It's the first result that'll come up. And this, just like you search PubMed for articles, you search the mesh database for terms. So in this practice where we're trying to find mesh terms, what you do is you take the term for the concepts you've identified from your research question, and you type them into the mesh search box, which, if you remember the old PubMed, this interface looks very similar to the old PubMed. So just typing, for instance, in vitro into the mesh search, you get a number of results, including the first result, which is in vitro techniques. And then once you arrive at the mesh record, you can use the PubMed search builder in order to capture the term and then add it to your search. So you can click on the add to search builder button in the PubMed search builder. It then populates the box there with the term itself, fully formatted and appropriately, including quotation marks and the field tag for mesh, which we'll talk about a bit, what field tags are and why they're important. But basically, this is the fully formatted mesh term that you can then copy, paste, and put into your search. There is a way to search PubMed directly using the Search PubMed button here. But we're interested in more than just this one term. We're interested in a whole string of terms to talk about in vitro techniques. So we're going to build our search offsite in our Word document and then put it into PubMed when we're ready to fully search it. So what that looks like in terms of your search documentation is under your first concept of in vitro studies, you can put this mesh term in. So that's your first term. And at this point, we find keywords. So there's a question, given that mesh terms are so awesome and capture all this heterogeneity and are uniform and systematic, why would we even need keywords? So we'll talk about that. We'll talk about where you can get keywords and then how they're added to your search. So, right, it's a good question. If mesh terms are the preferred terms that PubMed uses, why are keywords even needed here? And there are a number of reasons. One is it takes time for mesh terms to get assigned to papers. So papers are pouring into PubMed from publishers automatically all the time. And there are human indexers that are doing the work of assigning mesh terms. As I understand it, there's machine learning or machine-aided tools that help these humans, but nevertheless, there is a delay. And so if you just use mesh terms, you're going to miss the most recent scholarship on a topic. Also, I think, and I alluded to this earlier, new phenomena, assays, biomarkers, disease states, etc., may not have mesh terms yet. So mesh is a growing document or a growing system. Mesh terms are added every year, but there's a sort of deliberative process that goes into adding terms to mesh. And it's not an instantaneous, there's not an instantaneous assignment of a mesh term. And PubMed indexers just may make mistakes. So it's always good to have keywords as a backup in case the indexer reads the paper but doesn't think it rises to the level of a main idea, and so it's not going to assign a mesh term to it. You would capture that with a keyword. So finding keywords, one of the sources for keywords, as I mentioned before, is the mesh record. So mesh records are awesome for finding the preferred terms, but they also have these entry terms, which are ways of telling the indexer, hey, you see these terms out in the literature, this mesh term is probably appropriate. for that paper. But let me back up a second and just say that the mesh term itself should always have a keyword equivalent. So for instance, in vitro techniques, we're going to definitely include that in our search. There's a caveat. Sometimes mesh terms have a last name, first name format. So for instance, depressive disorder, comma, major is the mesh term for major depressive disorder. But when using keywords, you're looking in the title and abstract, and authors are not going to refer to their depressive disorder, comma, major patients. They're going to refer to major depressive disorder patients. So you want to just flip that when you convert that into a keyword. As I said, entry terms are very helpful lists of terms that can be used as keywords. Essentially synonyms for the concept you're interested in. There tends to be some redundancy, a fair amount of redundancy in the entry terms list, and also some of that last name, first name formatting. So you have to sort of do some cleanup. In fact, for the in vitro techniques entry term list, there's really only one keyword I would pull out of that, which is in vitro, because in vitro is part of every element or every bulleted term here. So that's all we need is in vitro. Any mesh terms that are child terms of a parent term, so for instance, organ culture techniques is a child term of in vitro techniques, you'll want to look at the entry terms list and pull out keywords from there. And so, for instance, organ culture would and its plural form would be good keywords to pull out of that list. Another source for keywords is on-target articles. And this is, again, I think pointing to the iterative step 8 in the process, where you have a dialogue between your search results and your search strategy. And there's always going to be changes. I won't say always going to be, but until you sort of reach your final search strategy, there's going to be change, oftentimes based on the articles that you're reviewing. So you'll find articles based on the terms that you've generated, but there'll be some terms as you read the papers and say, wow, this is a really good paper, you'll notice some terms that you hadn't included. And those will probably, should probably be included in your search in order to get more results like the one you're seeing and to broaden the net you're casting into the literature. So what you're seeing on the right-hand side is the Pubmed record that shows potentially relevant keywords in the title, abstract, and author-supplied terms. And so the keywords underlined in blue were used in the search that's found list. So when I did my a very initial draft search, all I had was a term for serotonin, in vitro, and major depressive disorder. And this paper hit all those features, or hit all those terms, and so came up in my results. But as I looked at it, I saw that the underlined terms in red should really probably be included in my next iteration of this search. So terms like patient-derived neurons, and induced pluripotent stem cells and transdifferentiation are pointing to in vitro techniques that really might be valuable. 5-HT, oh, that was already found. I had that synonym. But what I didn't have was serotonergic. And so that's a word variant that probably I should include as potentially useful. And then also this paper is looking at more than just major depressive disorder. And so this goes back to our, actually back to our research question. So iteration can go all the way back there to step 1, where you might say, I'm not finding very much on major depressive disorders. Maybe I'll expand this a bit to other psychiatric disorders. And so that's just some things to think about. Maybe this would be put in notes to say, consider adding mood disorders and bipolar disorder. The same paper has mesh terms assigned to it. So not only can you get keywords from the title, abstract, and author supplied terms, but you can also take a look at the mesh terms for an on-target paper and find some useful things there, mesh terms that you might not have included. So these are, again, standardized terms used to describe specific phenomena in PubMed, and you just may not have thought of these, particularly in the case of alternative approaches. So 3Rs approaches, this can be a way to uncover that kind of, scattershot, not scattershot, but just the fact that 3R terms are really spread fairly widely across mesh. Using on-target articles can help reveal that sort of hidden structure. So for instance, there's a neurological models mesh term, which you might not have thought of, which is sort of theoretical models for looking at nervous system function. Again, there's cell transdifferentiation and induced pluripotent stem cells. So those are keywords we saw, and they actually have equivalent mesh terms. So that's going to add more results to your list because you're using those standardized terms. And we had noted serotonergic as an adjective form of serotonin, and there's actually a mesh term on the neurons themselves. So that could really be helpful to include that bit of the biology in your search strategy. So yeah, just to reiterate that you want to look for the mesh parts of the PubMed record. And again, the ones in blue is what we use to find them. So cell culture techniques is part of the in vitro techniques hierarchy. So that's why that was pulled up. Major depressive disorder, again, in last name, first name format, that has a mesh term, as well as serotonin. And then I already went through the red ones that looked relevant. There's I didn't mention this, but there's a bipolar disorder mesh term. Don't see one for mood disorders, but there may be one. So a lot of times if I find a novel keyword, I'll go ahead and do a quick mesh database search to see if there's an equivalent mesh term, just to be sure. Okay, so in terms of adding keywords to your search, basically what I'm doing is I'm leaving space here after the mesh term to indicate that these are separate, distinct terms: in vitro culture technique, organ culture, and co-culture. And there are a lot more. So the hierarchy for in vitro techniques is quite extensive. So there'll be a fair number of additional entry terms to include here, as well as what I haven't shown here is anything about pluripotent stem cells or the other sort of in vitro things I just discovered from that on-target article. So the little ellipsis here is meant to indicate that there are going to be definitely additional keywords. I like to think of what we've talked about so far as the sort of conceptual steps of creating a systematic search. And now we're going to talk a bit about more mechanical steps, which, although I'm calling them mechanical, they're very important, even though they might seem, and maybe indeed are, quite mundane. They are very important for the functioning of the search. So you have to, for instance, in step 4, apply the appropriate formatting so that PubMed understands exactly what you're trying to do with your terms, give PubMed the proper instructions, and use the appropriate syntax so that PubMed recognizes the terms and what you're trying to do with them. So we'll talk about field tags, which I mentioned earlier. We'll talk about quotation marks for phrases, truncation as a way to generate word variants, and then how to actually add the formatting. So I mentioned field tags already, but I didn't really define them. They're essentially just sort of shorthand for telling PubMed where in the PubMed record you want to search for terms. And this is important. If you can put in a completely unstructured search in PubMed, say just a few terms without any quotes or any field tags or even any Boolean operators, PubMed will take that and try to figure out what you want to do with it. And one of the things that PubMed will do is apply what's called an all fields field tag. Basically, what that means is it's going to search for that word in every part of the PubMed record. So that includes author names, affiliations, journal names, fields that really may generate a bunch of off-target results. So knowing field tags and how to use them is really important in focusing your search. And we've already encountered them because when we generated our mesh term from the Pubmed search builder in the mesh database, it automatically applied the field tag of mesh to the term, which means that we're searching for that term in the mesh part of the record. So in the case of keywords, they look the same brackets, and then they just have various conventions. So TIAB, it stands for title and abstract. So using that field tag, we'll search in the title and the abstract of the article. TW stands for text word, and using that bracketed field tag, we'll search for title abstract as well, but also mesh, although not sort of looking for child terms, just the term itself, and then author supplied keywords. So TW, using TW is going to be more comprehensive in general than TIAB. And there is a complete list of field tags. Pubmed does a great job of documenting this in its user guide. So I provide a link here to the search tabs. And if you just go to the Pubmed user guide, which is linked, I think, off of every page in Pubmed, just go to the appendix and there'll be a list of all the field tags, what they mean, and how to format them. So super useful. You can focus your search down to affiliations, down to journal names, down to specific authors, tons of ways you can focus your search. Quotations are important. So they ensure that your phrase that you want is not split into its component parts. So a good example would be animal use alternative. That's an entry term under the animal use alternatives mesh term. And if you don't put quotes around it, Pubmed might split it into animal and use and alternative. So all three of these words are commonly used in multiple contexts. So if you split them apart and have them be searched in the abstract, for instance, instead of searching them as a phrase, very likely you're going to get off-target results. Just remember quotations for phrases. And then truncation is really handy to find variants. It saves time. Sometimes your list of synonyms that go into your concepts can be quite long. So limiting your number of terms by using truncation can help you avoid errors, which can just naturally occur the more terms you add. So in the case of Pubmed, truncation is used in a lot of databases. So the rules for truncation might be different in other databases, but in Pubmed, use an asterisk. It has to be at the end of a word or the end of a phrase. So you can't put it in the middle of a word or at the beginning of a word. It has to be at the end. So for example, co-culture with an asterisk will find the plural form as well as co-cultured. And then tissue culture, where you put the asterisk inside the quotes, we'll find, again, the plural form and then tissue cultured as well. I find a lot of times that these truncation can actually find variants I didn't even think of or didn't know existed. So it's handy for broadening your search. Just be careful not to use it for too short of a word. So just use caution. Don't use it for like a common like 5-word prefix or something that might give you off-target results. So in other words, just think carefully before you apply that asterisk to your term. So in terms of adding formatting, it's fairly straightforward. You just look at your keyword list. You see any phrases, put them in quotes. And if they're keywords, you apply the TW field tag. This is kind of what that looks like. And then our next step is also a sort of a mechanical or process step. And it's very straightforward. It just involves adding Boolean operators to tell Pubmed how you want to combine your terms. So we're going to talk about 2 operators, or an and. There is a not operator, but I tend to discourage using it because you can inadvertently exclude articles by using not. It's kind of a rough tool in a search. It can lead to unintended consequences. I'll just say that. So the or operator is used between terms within your concept. So it's used to connect synonyms, essentially. And what you're telling Pubmed is that I'm interested in any of these terms, in vitro or culture technique or co-culture. And then for the and operator, this is where that abbreviation or sort of shorthand comes in handy. So this is a somewhat fleshed out document showing all of your concepts with terms included underneath them. And they're all the concepts have ORed synonyms in them. And then instead of trying to do the potentially error-prone work of putting parentheses around concepts and then anding them together, I just say concept 1 and concept 2 and concept 3, indicating I'm interested in the intersection of all 3 of these concepts. So now we move on to sort of the assembly, running the search again, which is a sort of mechanistic process. And what we're going to do is allow Pubmed's advanced search feature to do the anding together and do the putting of the parentheses together. So we'll talk a bit about the advanced search builder, how I like to run concepts one at a time in order to check for search errors, how you then use the search builder to combine or assemble the complete search and run it. So the advanced search builder, there's a link to the advanced search from the Pubmed homepage, just underneath the single search box. And it looks like what you see at the bottom of the screen. And we will be using the query box because we have assembled our search off-site. There is a way to sort of do searching on the fly using the box up at the top where you're adding terms. But we're not going to do that because we've done it offsite. So I like to run concepts one at a time in order to see if there are any mistakes. It's much easier to detect mistakes within concepts than trying to figure out in the fully assembled search what went wrong. And in fact, some of the errors that you might have otherwise detected can go missing in the broader search. So for instance, here I've added just the serotonin concept and I put that in the query box. I click on add to history, which is a drop down from the search button in advanced search. Just add it to the history and that enables me to take a look at a couple of things. The results, the total number of results, as well as the search details. So in a dramatic instance, if you did a depressive disorder, major depressive disorder search and only found, say, 130 results as opposed to 13,000, then you'd know there was something wrong, like you misspelled a word or you left out a Boolean operator or something. So you can use the results number as a gross check for errors. Ballparking, like, okay, that number looks reasonable. But I also recommend taking a look at the details. So if you click on the down arrow under details, you can see exactly how Pubmed has translated your search. And in this case, what we find is that between the mesh term and the first keyword, we left out a Boolean operator. There is no or. And so because of that, Pubmed automatically converts any space between terms into an and. So this 13,000, even though it looks big, is actually a truncated version of what it should be. So what we should have found, if we take a look at the very bottom of the screen, here we have inserted an or that should have been there between the mesh term and the first keyword, and we see we get 41,000 results. So that's a big difference, 13,000 and 41,000. So again, even though these are sort of mundane things that we're doing here, somewhat rote, they are important. So it's good to have a good attention to detail and to use the search details to detect any possible errors. So it's really simple to assemble this once you run the individual concepts. There's an actions feature with a little ellipsis, and you click on that and you can add the first concept, which is in vitro, using the add query command. And then after that, every subsequent concept can be added also using the actions feature, using the add with and command. Since we're anding all three of these, that's the one we would use for serotonin and major depressive disorder. So then what appears in your query box at the end of that process, once you've added all the concepts together, is that work that has the real potential for error, where parentheses have been inserted in the appropriate places to delineate the specific concepts, and then the Boolean and has been inserted again between the concepts as we want it to. Once that is all done, then you click the search button and get your search results. And then you can do your 7th step, which is assessing search quality. And so we'll talk a bit about sort of the results as a number, as sort of this a gauge of quality, and when I say quality, I'm a lot alluding to comprehensiveness, but also alluding a bit to precision. So in any quality search, you want to balance between the two. So you want it to be finding what it should be finding and what you know it should be finding, but you also want it to be have a measure of precision so that you don't have to look through 20,000 articles in order to find what you're looking for, right? So we'll talk a bit about the proportion on target, sort of that precision measure, and then we'll talk about testing against a gold standard set, which really gets at recall. So this is what your Pubmed result looks like. We only found 90 results from this really early draft search. And that's a bit of a red flag. It could be that really there are only 90 papers or 100 or so papers on this topic. This really might capture the scope of it. But it's more likely that we need to do some more work. So we need to iterate the search and add some more terms based on the on-target results we're seeing in the result set we get. I want to point out, in addition to pointing out, that you can see numbers of results, that you also get some error messages on the result page. So you might see this quoted phrase not found in Pubmed, and that's handy, should be paid attention to make sure you're not misspelling a term. In this case, these just were spelled correctly, they just were not found. They were entry terms that were not found, which sometimes happens. So this won't hurt your search, but if it's a misspelled term, obviously that is a problem and should be corrected. So 2 other ways, as I wrap up here, 2 other ways to assess search quality. One is to look at a sample of results and see what proportion are on target. So kind of getting at that precision measure. So a lot of times what I'll do is I'll set the display options for Pubmed to show me 100 results at a time. I'll also format it to show me the abstract view. And then I'll be sure that I'm logged into my NCBI account and have set up highlighting for terms. And that way I can easily see in my results where the terms are hitting. And that just helps me assess whether the results I'm seeing are on target. So if I do 100, then that's an easy way for me to say, okay, 20% are on target, or 50% or whatever. And sort of a rule of thumb is if you get 99% are on target, chances are that you could do a more comprehensive search. I'm not saying that you have to, but that depending upon what the uses you're using the search for, if you're wanting to make sure you haven't left out something relevant, then probably a 99% on target rate means that your search is too focused. On the other hand, if it's like 1% or 5% or 10%, then that is probably you've got too broad of a search. You need to focus it somehow, or maybe you've got terms that are blowing it up and giving you off-target results. So that again gets at the iteration, importance of iteration. What's really valuable as well is to have a sort of pre-established set of on-target or gold standard articles. So these could be papers you've been gathering over the years that you know are like really on target for the research question you have, or might be the product of a preliminary search where you just throw in a few keywords or maybe get a few review articles and see what these review articles have identified. Having those, which might be like 10, 15, 20 articles that are on target, having those is golden. Pardon the pun, but what you can then use that for is sort of an internal check. So you can run those articles as a search, and I typically do it as like a list of PMIDs. And then you can run your search that you think is maybe finished, and then you can add those together and see if you miss something. Because if you don't find all the gold standard articles with your search strategy, then you've made some error and you need to figure out why. So you can look at the articles that you didn't find. Sorry, that's my timer. You can look at the articles you didn't find and say, well, what terms do I need to include from these articles in order to find them? And I am wrapping up. So I just have a few more slides here. Yeah, talking about the last step, which is iteration. So I have been talking about iteration practically the whole time, but just to reiterate iteration, building a comprehensive search really isn't done in like a single 2-hour session, on one day. It's iterative. It takes some time and versions to kind of get to the final product. And so, as I've mentioned before, examining on-target articles for additional search terms is really important, as is looking at off-target results and seeing, what terms maybe should be removed. And so in iteration, you basically do that at each version. You make changes, you run the search again, and you repeat until the search demonstrates a good balance of precision and recall, and sort of uses those measures that we talked about of assessing search quality. You can sort of say, okay, I'm getting a certain proportion on target that I'm happy with. And also I'm finding all my gold standard articles. That would be sort of a good place to say, I think I've done enough and I think I can stop. So just wrapping up, I want to mention a few resources. So my library, Welch Medical Library, has created some great YouTube tutorials that cover a lot of what I covered very briefly today in much more depth. So I highly recommend you taking a look at that playlist. I've also created a Prezi on advanced PubMed searching, which again, sort of talks about these things just sort of in a different way, in a different format. So that can be handy. And then I provide some links to the Cochrane Handbook, the chapter on searching, and a link to the ECVAM search guide, which I really only talked about as an inspiration, but I think is really worth taking a look at. One of the things they do is provide sources for finding 3Rs types of studies. That of course includes PubMed, but includes a lot of other sources. So I recommend it for that. Also recommend it because they have sections on controlled vocabularies and terms. So controlled vocabularies and terms that are specific to 3Rs-based topics. That can be a great place, great resource to mine as well when you're sort of just getting started and trying to figure out what terms to use.